with this. Thank you very much for joining us, and I'm sure we'll have wonderful experience in the coming 4 hours. I would now hand over to Dr. Shambhu. I am Dr. Shantanu Tripathi. I'll be there as much as possible through this whole period of 4 hours. So thank you again, and I'll now hand over to Dr. Shambhu Samrat Samazdar to start his introductory session. Dr. Shambhu, please.
Thank you very much, sir. Good afternoon, all respected delegates. And it is always honor to talk on good clinical practice in front of Professor Shantanu Kumar Tripathi, sir, because he was one of the pioneers to implement good clinical practice in India. So today, for next 30 minutes, we'll discuss about on these broad headings. That is to understand the definition of GCP, how GCP evolution had occurred, what is the purpose of GCP, and the principles of GCP. And at the end, we will discuss about some points regarding the pre-marketing trial phases. So what is good clinical practice? Good clinical practice, this research word is not there. So it is actually good clinical research practice. And if we go by the definition given by ICH E6, then this is a standard for the design, conduct, performance, monitoring, auditing, recording, analysis, and reporting of the clinical trials that provides assurance that the data and reported results are credible and accurate, and the rights, integrity, confidentiality of trial subjects are protected. So this is a standard for the different parts, and this ensures two things. Number one, the results are credible and accurate, and number two, rights of the subjects or research participants. So there is another definition given by WHO in 1995. This is a standard for clinical studies which encompasses the design, conduct, monitoring, termination, audit, analysis, reporting, and documentation of the studies, and which ensures that the studies are scientifically and ethically sound, and that the clinical properties of the pharmaceutical product may be diagnostic, therapeutic, or prophylactic that under investigation are properly documented. There is one important vitamin M, rather we can say two vitamin M is important for good clinical practice. This vitamin M is not money. This vitamin M is morality and materialism. So good clinical practice or ethics is generally a multiplied product, we can say, of morality and materialism. If in a study the morality is 0, high materialistic value, but ultimately then the ethics will be 1 into 0 or 5 into 0 or 100 into 0 is equal to 0. Side by side, if the study has a very high ethical or we can say morality, the morality standard is very high, but there is a 0 materialism, 0 science, then ultimately again it will be the ethics is equal to then 0. So ethics is a combined product, or you can say a multiplied product of both materialism and morality. So while talking about evolution of GCP, we have to consider different atrocities that happened to the mankind due to this clinical research. So we all know this diagram that was taken from the Nazi war crimes, when the prisoner were subjected into very cold water, ice cold water, and they are observed for at least 5 hours until they die. So these are the examples of tremendous atrocities, And we all know about Tuskegee syphilis trial, where only the black African prisoners were selected to see the natural history of syphilis. No treatment was given to them. Thalidomide disaster, that was a very important one, that the mother was given thalidomide to treat their motion sickness, and what happened, there was tremendous atrocities in form of their children. So seal like deformity happened into their children. So next, the Jews chronic disease trial, Willowbrook study, Milgram study, these all are the examples of different unethical research works. And with the response to that atrocities, so we all know that the knee-jerk response, if there is some effect, there should be some counter effect, and to have that counter effect, there is evolution of different ethical principles. So these are the different deadline. The Nuremberg Code, in some, I think it is 1948, not 1949. Then Declaration of Helsinki 1964, the Belmont report, and ultimately the SIOMS, that is the Council for International Organizations of Medical Sciences, which developed 1993. So these three are the very important structure, or we can say the triangle of ethics. The Belmont report 1979. The first term is the autonomy of the individuals. That means the respect for persons, specifically to protect for those with diminished autonomy. Next is the beneficence. So how to maximize the possible benefits and minimize the possible harm, that was taken care on this beneficence principle. And the justice, that subject should be selected fairly to ensure that the benefits and burdens of the medical research are evenly distributed. So there will be an equitable distribution in the community with both the benefits and the burdens. So the autonomy means each person should be given the respect, time, and opportunity necessary to make his or her own decisions. Beneficence obligates the researcher to secure the well-being of all study participants. Belmont 1979, maximize the possible benefits and minimize the possible harms. So this was this was included in the Belmont report 1979. And justice, already we had discussed about that equitable distribution of benefit and harm. So it is a difficult and complex ethical issue. The attempt should be made to distribute the risks and benefits fairly without any bias. Who should bear the risk of a such study and who should receive its benefit? The participants not be selected due to class, socio-economic status, or race, unless that is justified by the study objectives. So 1990s, that decade is the decade of harmonizations in the field of clinical research. And we all know that in the 1990s, when we were child at that time, we were in love of Tintin, and the Tintin belongs to the place that is Brussels, and that Brussels in 1990 International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use, that was fast held. And we all know that Tintin was very much ethical in all aspects. So from that conference, there was something happened which helped us to be more vigilant in the field of clinical research, to maintain good clinical practice conduct. And the main participants or primary participants were USA, European Union, and Japan. And there were some regulatory and industry representatives were also there. Observers from Canada, Australia, and Nordic countries were there, and WHO acts as a facilitator. So what is the purpose of GCP? What is the purpose of good clinical practice? There is two important cornerstone. One is the quality data and another is ethics. And the data that reported results are credible and accurate. That means if it is a credible data or accurate data, that means it is a quality data, and rights, integrity and confidentiality of the trial subjects are protected or not, that has been taken care of by the ethics. But again I want to repeat that thing, that ethics is equal to a multiplied product of materialism and morality. So a bad science is also a bad ethics. So these are the general principles or 13 principles of GCP. Number one, ethical conduct. Two, risk benefit assessment. Subjects over science and society. Adequacy of pre-existing information. Scientific validity. Independent review. Medically qualified care. Trained and experienced personnel. Informed consent. Efficiency of information handling, confidentiality, drug accountability, and throughout quality among the systems and procedures. So number one principle, that is the ethical conduct. So clinical trials to be conducted in accordance with the ethical principles. Already we had discussed about the basic ethical principles, that is the autonomy, beneficence and justice, as per the Declaration of Helsinki, and that are consistent with GCP and the applicable regulatory requirements. Next is the risk benefit assessment. So before initiating a trial, weigh foreseeable risk and inconveniences against the anticipated benefit for the individual trial subject and society, and initiate and continue only if the anticipated benefits justify the risk. And this is also very much time dependent, research dependent, research objective dependent. So in presence of, just an example, in presence of COVID-19, we are also allowing some drugs which are not undergo or which don't have any that much relevant data. But in other situation, if I want to treat hypertension patient, so we should have much more robust data, much more data on phase 3 clinical trials before prescribing that drug to a hypertensive patient. So subjects over science and society, this is again very important. So the rights and safety and well-being of the trial subjects are the most important considerations and should prevail over the interest of science and society. So it is not just like that I want to give one or bring one anti-COVID vaccine, and I want to give it in some subjects, and I don't know how this vaccine will act to that subject. I am not convinced with the safety from the animal studies. So there is nothing to start or nothing to recruit those persons to have that vaccine trials, because first of all I don't want to put the science and society ahead of that particular subject. So subject safety is most important, subject's well-being is most important. So adequacy of the pre-existing information. So the available non-clinical and clinical information on an investigational product should be adequate to support the proposed clinical trial. So if I am going to start one phase 2 clinical trial, I have to be convinced with the preclinical data, phase 1 clinical trial data before starting the phase 2 clinical trial. So we have to be very much convinced with the adequate pre-existing information. The scientific validity. So clinical trial should be scientifically sound and described in a clear, detailed protocol. So bad science is bad ethics. Independent review. A trial should be conducted in compliance with the protocol that has been received prior institutional review board or independent ethics committee approval or favorable opinion. So this is again important, and that is why the institutional ethics committee are so much important. Medically qualified care. So the medical care given to and medical decisions made on behalf of the subjects should always be the responsibility of a qualified physician, or when appropriate of a qualified dentist. So this is very important, the qualification of the physicians, because it is not like that I am participating in clinical trial means there is I should also get the optimum or the best treatment which should be provided to me. So trained and experienced personnel. So each individual involved in conducting a clinical trial should be qualified by education, training, and experience to perform his or her respective task. And this is why this GCP training and GCP certificates are also very important, because this is a dynamic process. We have to well known about the concepts of good clinical practice. Informed consent, freely given informed consent should be obtained from every subject prior to the clinical trial participation. Efficiency of information handling. All clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. So we all already discussed about the data credibility. So this principle helps us to have a data which is credible. The principle applies to all records referenced in these guidelines, irrespective of the type of media used. Confidentiality. The confidentiality of the records that could identify the subjects should be protected, respecting the privacy and confidentiality rules in accordance with the applicable regulatory requirements. Next is the drug accountability. Which drug I am going to test it? That investigational products should be manufactured, handled, stored in accordance with the applicable good manufacturing practice. They should be used in accordance with the approved protocol. So next or last principle of GCP, that is the quality through systems and procedures. So systems with procedures that assure the quality of every aspect of the trial that should be implemented. Aspects of the trial that are essential to ensure the human subject protection and reliability of the trial results should be focus of such systems. So these are the basic principles of GCP, and that is why it is always taught to us that when we are going to start a clinical trial or we want to start one research, then you think yourself as a research participant and assess whether it is ethical or not. So do unto others what you would like others to do unto you. And that is why our ancestors told us the same things 4,000 or 5,000 years back. Atmana pratikulani pareshuna samacharet. And this was about the GCP, and I will just touch upon some important points regarding the pre-marketing trial phases. So before going to pre-marketing trial phases, I just want to focus on few issues, that what is clinical trial and what is not, because there are some confusion always in our mind. So this is an algorithm to understand which type of clinical research we are going to conduct. First of all, did investigator assign exposures? So if I am investigator and I am assigning that these people will take this drug and these people will take this drug, and I am going to observe what happens. So that is that if that is yes, so I am doing one experimental study. If it is no, that means I am observing. The other people is assigning the exposures, I am observing the outcome. So that is the observational study. Then if it is random allocation in case of experimental study, then it is, if it is yes, then randomized control trial. If it is no, then non-randomized control trial. And for observational study, whether it is a comparison group, if we want to compare, then it is an analytical study. Then if it is not comparison group, then it is a descriptive study. And if it is an analytical study, then we have to check the direction. That means if we are starting from exposure to outcome, then it is a cohort study. If we are going back from outcome to exposure, then it is a, that is a retrospective one, then it is a case control study. I am telling it, it is in a simple way, but there are some exceptions also in these things. We are not going in details on that. And if the exposure and outcomes are at the same time, only one visit we are taking the data, that is a cross-sectional study. So what is clinical trial? So as per the new drug clinical trial rule, which came upon 2019, any systemic study of a new drug or an investigational new drug in humans to generate data for discovering or verifying its clinical, pharmacological, that includes pharmacodynamic and pharmacokinetic or adverse effects, with the objectives of the determining its safety, efficacy or tolerability, is called as a clinical trial. So before going to the phases of clinical trial, just one point, because there are some area of drug discovery which is preclinical, and that drug discovery which is a preclinical one, that is noted as target identification or validation, then some assay development, then to find the hit, from hit to lead, then lead optimization, then ultimately the candidate selection. That candidate will go into the preclinical stage, and in preclinical stage, this is a funnel we can see here, that when start with the from the hit to lead, and there is before that the high throughput screening, there is more than 1,000 compounds are there. From that it becomes hundreds, then optimization done, then some 12 to 20 compounds are there, then candidate seeking process, ultimately it be narrowed down to 1 to 3 compounds, then the preclinical development happens. And in the preclinical studies, there are broadly divided into two groups. One is pharmacokinetic and pharmacodynamic studies, and another is preclinical safety studies. Among the pharmacokinetic pharmacodynamic studies, we have to evaluate the effects like pharmacokinetic changes, absorption, distribution, metabolism, and elimination. How about the tissue distribution? Whether the mechanistic studies are feasible or not, we have to check. In case of preclinical safety studies, there is a broadly safety pharmacology and toxicology, and there are some different toxicology studies. These are not to be discussed in a short time. That is a separate chapter. So what are the phases of clinical trial? After having the preclinical studies, then we have to enter the phase 1 clinical trial. So that generally for the objective is to understand the safety of the drug, to have some idea the dose which should be used first in human. So first in human dosing, and the primary objective is to determine the safety. Very small group of participants, around 20 to 100 participants were taken. Then it becomes the phase 2 after doing the phase 1 clinical trial. For phase 2 clinical trial, again the primary objective is safety, assessment of the safety and the dosing. So we have to find the dose which is effective. Then the participants little bit more here in the phase 2 clinical trial, around 200 to 400 participants were taken. Then from phase 2 to phase 3, the main objective is to assess the safety and efficacy. Here the research participants number is much more, from 1,000 to 5,000 like that, and we all now know about the phase 3 clinical trials of different COVID-19 vaccines, which includes 20,000, 30,000, 40,000 population. And after that, the DCGI will check whether it can be approved or not. Then after approval, there is another trial, that is the phase 4 clinical trial, and that generally for the post-approval surveillance. And the main goal for that is to assess the safety and efficacy in a larger populations. So there is two important terminology again. Before moving into the phase 1 clinical trial, we have to apply for IND. That means investigational new drug application at that time. And after having the phase 3 clinical trial, when we are providing or we are giving it to the DCGI, then we have to apply for NDA. That means new drug applications, that is submitted for DCGI for approval. And these are the broad headings what clinical pharmacological plan in the drug development. These are the broad tests for safety, pharmacokinetics, and mechanism of action understanding. These are the tests like SAD, single ascending dose, multiple ascending dose, proof of concept or proof of mechanism understanding, mechanism of action understanding, pharmacogenetics and pharmacogenomics studies, QT studies, QT toxicity studies. Then for biopharmaceuticals like food drug interaction testing, BAB studies, that is bioavailability, bioequivalent studies, some line extension studies, detailed absorption, distribution, metabolism, and excretion elimination studies. There is a provision in the phase 3 clinical trials generally to see, or sometimes phase 2 clinical trials also, to see the effect of the drugs in special population, safety of the drugs in the special populations like renal compromised, hepatic compromised, pediatric population, age or gender, different age groups and different gender patient populations. And also there are multiple drug interaction checking also done in the different phases of clinical trial. So here we can see in a nutshell where different subgroup of clinical pharmacological experiments done in the different phases of clinical trial. So this is the way, or this is the path how the new drug came into the market, from the concept, from the target identification, target validation, and hit identification to lead selection, then lead optimization, then a new chemical and entity identification. That will go under the preclinical trial, and after having the preclinical trial convincing data, they will apply for investigational new drug application. Then that investigational product came into the clinical trial. Clinical trials, that means phase 1, phase 2, and phase 3 clinical trial. If they are convinced, they will apply for new drug application, and the new drug will come into the market. So this was my last slide, and I am very thankful to this particular book, which was written by my colleague Dr. Shaugato Sarkar. Very interesting book, and I found it will be very helpful for all the postgraduate pharmacologists, even those who are have some interest in the field of pharmacology. So this is one question I want to raise, and it can be discussed later when Professor Shantanu Kumar Tripathi sirs will have a doubt clearance session. So as a ethics committee member, will you allow this study? So this was a published literature, development of a simple radiant heat induced experimental pain model for evaluation of analgesics in normal healthy volunteers. So which analgesics were tested here? It is tramadol, and we all know that tramadol is a well accepted drug for last 20-24 years. It has a pain reduction property. So they prepare one pain model, and they give this tramadol to see how this is effective in reducing that pain in normal healthy volunteer. So my question is that as ethics committee member, keeping in mind all the principles of ethics, whether you will allow this study, if they are this is submitted to your ethics committee or not. So I want to end with this important sloka, and I think this is one of the important slokas that we should, all the researcher at the beginning of the research should remember. That is कर्मण्ये वाधिकारस्ते ma phaleshu kadachana ma karma phal heturubhu ma te sangas takarmani. And in simple, it is the no attachment to outcomes, nato, no attachment to outcome. So when we are starting our research, we should not be worried about the outcome. We should perform the research, and this is important. While talking with our research participants, we should be very much clear to them that as I am giving you this exposure, this intervention, I am also not aware what is the outcome, because there should not be any false belief among the research participants that I am treating them, so that treatment is going to benefit him surely. So with this, I want to thank you. Thank you, InnovoCare Health Hub Solution, for giving me this opportunity to be in front of the august audiences, and specifically to sit in front of, though this is a e-platform, but via this e-platform also, it is, I am very much thankful to you to give me this opportunity to sit in front of my guru, my professor, Shantanu Kumar Tripathi sir. Over to you, sir. Sir, can you hear us?
Now I'm I was muted actually.
Oh.
Now I'm unmuted. Okay. Okay. Thank you very much, Dr. Shambu. It was you have kept in time, and very otherwise nicely you have elaborated on the principles of good clinical practice. And I particularly like the way you completed it. And the last slide, as you mentioned about that no attachment to the outcomes, I think consciously or not so consciously, you have really pointed out that one of the basic principles of ethics, and that is, okay, not trying to be biased. Okay, as researcher, all of us have biases. Okay, and a given intervention that as an investigator I am trying in a clinical trial, it is very, very likely that I get a little bias towards that intervention, the intervention that I am trying actually. So most of the scientists, okay, it is very common that the scientists, they consider their invention as almost their baby, and they would always try to okay, demonstrate that how great the invention is or how good the invention is. Okay, they become emotionally attached. But now you say that no, there should not be any attachment. And basically this good clinical practice and the ethics principles that actually teach us that how we can remain objective, how we can distance ourselves from the outcome, how we can consciously teach ourselves that irrespective of whether a given intervention ultimately, I'm talking of experimental research, of course, clinical trials, interventional outcome, whether or not it succeeds, okay, it becomes efficacious. Okay, I should have equal kind of feeling that if it succeeds, it's great. If it doesn't succeed, I should not actually be get frustrated or get disappointed. So the failure in clinical research also has a lot of lesson for us, so that we can try to learn from that and also plan the next research, okay, where we can think of a success. So success or failure, the researcher should always be committed to their principles, and they should lay their hands into their research project with equal kind of passion or equal kind of uh, concentration, attention. So having said this, I think there might be also opportunity for discussing this further. Particularly, I would like to also get back to that definition of good clinical practice, which is a extremely loaded, you can say, paragraph. When you say that the way the good clinical practice has been defined, but broadly speaking, it has two components. One, the data credibility component, that whatever data we generate through clinical research, it should be credible. On the other hand, while doing so, because we are engaging ourselves into clinical research, which means we are uh, we are involving human subjects or human individuals uh, in achieving our research goals. Okay, so there is always an inherent ethical problem, and that has to be properly addressed. All right, so that is the second part, that is uh, maintaining the without otherwise jeopardizing, without otherwise compromising the rights of the human individuals who are participating in a trial, taking part in a trial. Okay, so we used to say earlier that the research is being conducted on these people. So that is to be, we have to come out of this kind of an understanding, and we have to rather say the research was conducted with these people. So it's not on these people, it is rather with these people. So when you say with these peoples, we otherwise accept the participation or the contribution made by these people. So the investigator and the research individual, research participants, we accept that they are on the same board. It's not that the investigator is a higher pedestal. But when you say research on these people, subconsciously we are putting ourselves as the researcher at a higher pedestal. Ethically, that is not sound. So we'll discuss this further. But now I think it is time to pass to the next speaker. And I would also request the members of the audience, if you have questions, comments, you can write down in the chat box, so that at the end of each session also we can